Hey people, it's Niles Toki. Now this is from the Daily Mail, and I like to do these science videos, so here's another one. Human genes are inserted into monkey brains, causing them to expand. It is unnerving Planet of the Apes style experiment. Genetically engineered marmosets found to have bigger human-like brains. ARHGAP11B gene was used to increase the number of neurons in neocortex. The gene caused wrinkled grooves like human brains to increase surface area. Scientists have inserted a human gene in the brain of monkeys to make them larger and more wrinkled in disconcerting lab experiments. Experts found the insertion of the gene called ARHGAP11B resulting in larger neocortex in the fetus of a common marmoset. The neocortex is the deeply grooved outer layer of the human brain that is involved with reasoning, language, conscious thought, and other important functions. The gene, which is found in humans but not non-human primates or other mammals, triggered the monkey's brain stem cells to form more stem cells enlarging the brain. Genetically tweaked marmoset brains were found to mimic the natural bumps and indentation in human brains, known as the gyri and sulci respectively, an evolutionary trait in humans to increase surface area for neurons, nerve cells. The experiments are evocative of the recent Planet of the Apes films where genetically modified primates wage a war on humanity. Researchers developed 7 marmoset fetuses in total, all in utero, inside the womb, that's in brackets, and were obtained at day 102 of pregnancy by cesarean section for analysis. We found indeed that the neocortex of the common marmoset brain was enlarged and the brain surface folded, said study author Michael Hayde at the Max Planck Institute of Molecular Cell Biology and Genetics, MPI-CBG. We also saw increased numbers of upper layer neurons, the neuron type that increases in primate evolution. The human neocortex is about 3 times bigger than that of our closest relatives, the chimpanzees. During evolution, our brains folded into the distinctive wrinkled appearance to fit inside the restricted place of our skull, while allowing the surface area of the neocortex to be greatly increased. Images of the 101-day-old genetically engineered marmoset fetus about 50 days away from its normal birth date showed its induced folding in the team's experiment. This contrasts with marmoset brains normally, which are much smoother than human brains as well as smaller. The gene may have caused neocortex expansion during human evolution, according to the team, which also include experts from Central Institute for Experimental Animals (CIEA) in Kawasaki and the Keio University in Tokyo, Japan. The Japanese researchers include Hideyuki Okano, had pioneered the development of technology to generate transgenic non-human primates. Okano's lab at the Riken Center for Brain Science in Wako, Japan was the first in the world to produce transgenic marmosets with germline transmission (GT). GT is a technique where embryonic stem cells contribute to the reproductive cells of a mammal germ cell and are genetically passed on to its offspring. However, there was no use of GT for this project for the simple reason that transgenic marmoset fetuses were not allowed to be born. We confined our analysis to marmoset fetuses because we anticipated the expression of this human-specific genome would affect the neocortex development in the marmoset, said the study author Wieland Hutter at MPI-CBG. In light of potential unforeseeable consequences with regard to postnatal brain function, we consider it a prerequisite and mandatory from an ethical point of view to first determine the effects of ARHGAP11B on the development of a fetal marmoset neocortex. ARHGAP11B arose through a partial duplication of the ubiquitous gene ARHGAP11A, approximately 5 million years ago along the evolutionary lineage leading to Neanderthals, Denisovans, and present-day humans. The new study, which has been published in the journal Science, follows work at MPI-CBG in 2015 to identify ARHGAP11B. Researchers isolated different subpopulations of human brain stem cells and identified which genes are active in which cell type. Tests at the time on mouse embryos revealed that the gene can have a huge impact on brain development. When expressed in mice on physiologically high levels, cause an expanded neocortex. Embryos injected with the gene grew larger brain regions, and some developed a wrinkled surface that's characteristic of the human brain. It's so cool that one tiny gene alone may suffice to affect the phenotype of the stem cells, which contributed the most to the expansion of the cortex. Study-led author Marto Florio at MPI-CBG told Live Science at the time. However, the gene's relevance for primate evolution had been unclear until now, researchers said. How our brains have grown. Around 3.8 million years ago, our ancestors Australopithecus afarensis had a brain that was 30 cubic inches, 500 cubic centimeters in volume. About 1.8 million years ago, Homo erectus had a brain twice the size of that one. When Neanderthals and Denisovans arrived, the brain had grown to 85 cubic inches, 1.4 liters in volume. Despite this increase in size, scientists believe humans' intelligence may have more to do with how brain cells form than how large the brain grows. Human-specific gene ARHGAP11B arose through a partial duplication of the ubiquitous gene ARHGAP11A around 5 million years ago, along the lineage leading to Neanderthals, Denisovans, and present-day humans. After this lineage had segregated from that leading to the chimpanzee. A sequence of 47 amino acids is essential for ARHGAP11B's ability to increase brain stem cells. Well, I hope you've enjoyed that. Please comment, rate, share, and subscribe. I'm going to leave this article in the description. If you'd like to support the channel, leave my GoFundMe there as well. If you'd like to follow me on Instagram, I'll leave that there as well. If you'd like to buy one of my t-shirts, I'll leave that there as well. Peace.